Good morning or good afternoon. Hey guys, and welcome to another awesome lesson on DNA and its translation. So I'm just going to briefly recap stuff that we've gone over so far to make sure that we understand where we are in terms of the context, and then we're going to go ahead and talk a little bit about transcription and translation, which is a real big topic for today. So let's go ahead and dive in. The last couple of times we talked about DNA structure, we said that it was the genetic material, and this was proven, or at least suggested, by experiments by Griffith and Avery and Hershey and Chase, who radioactively labeled proteins and nucleic acids to see which one was actually being injected into bacteria by viruses to replicate the viruses. They saw that it was in fact the nucleic acid or the DNA, and that provided some of the definitive evidence that yes, in fact, DNA is the genetic material. This led us to the structure of DNA, where we talked about Rosalind Franklin and her boss Wilkins, and then Watson and Crick, who were all able to put together some data on the helix in order to understand the structure of DNA, so they could understand how it replicated, how it worked, how it was able to create features of organisms, etc. They were able to use X-ray crystallography to bounce shadows and to create the famous photo 51, which shows that the DNA was a double helix, and we learned different things from this pattern about the structure of DNA. This is one thing we didn't go over because a lot of this stuff isn't wholly covered in your curriculum. Remember, this is an IB actually associated site, but I really like it, so that's why I'm using it. But the structure of DNA is going to be directly related to its role as a genetic material, because the structure of any molecule is going to tell you how it works, right? Proteins, their structure is long chains of these amino acids that have these different side groups that are going to bind in a certain way that should tell us that they're going to form into 3D structures. DNA is this repeating sequence of codes within a double helix. It should let us know that it's going to store information. So the structure of a molecule helps to determine its function. But there are some sequences that don't encode proteins, which I have mentioned before. These are non-coding regions of DNA, such as satellite DNA, short tandem repeats, telomeres, introns, non-coding genes, and gene regulatory sequences. Now, we have talked a little bit about these. The gene regulatory sequences are like promoters within transcription and stuff like that. The introns are what we cut out of the mRNA back again with post-transcriptional modification, which we talked about last time. Telomeres are just going to be, we haven't talked about these, but these are just caps on the ends of chromosomes to help to protect against DNA damage. Satellite DNA, short tandem repeats, these are just small repeating segments of DNA. And then, of course, non-coding genes are going to be the genes coding for tRNA and rRNA, because of course we have to make those somehow in our body, and so that's how they're made. So you can remember this by STING, but again, it's not wholly important or vital for you to know. Non-coding DNA, STR profiling. This is back when we talked a little bit about DNA profiling and gel electrophoresis and stuff like that, looking at the number of repeats and things like that. Again, I'm going to skip over this because the more important stuff is later on. So we talked about the organization of DNA, that most of the time DNA is not in a double helix. It spends most of its time in a very highly condensed and twisted form called chromatin, which makes up our chromosomes. But when DNA does need to be used, it needs to be unwound like yarn from a ball of string. And when it's unwound, we can go ahead and access the DNA. We know that certain environmental things can cause the DNA to wind more tightly or more loosely around the balls, making them more or less accessible. This is called our epigenome, is these molecules that make it wind tight or loosen, and that controls the expression of genes, which controls our traits in our bodies. Nucleosomes are where we call what we call it when DNA is wrapped around those little histone proteins, the little balls. Then we talked about DNA replication. We talked about the structure of DNA. It was a anti-parallel structure with the the bases in the middle and the phosphate sugar backbone on the outside. They're traveling in two different directions, from the 5 to 3 and the 3 to 5 direction. We talked about the different proteins that were involved in the process and how they were able to put together DNA and replicate it, basically copy it, and each copy would be inserted into each new cell that was made. And in the end, we end up with, of course, two identical strands of DNA. And we call it a semi-conservative replication because it's utilizing one half of the original strand to build the new strands. And so we don't have to completely recreate a new strand. Now, all the rest of this stuff is stuff that we didn't really need to talk about, so let's move on to the next little piece. So we then talked about transcription, and we said that transcription was basically where we go ahead and we take the gene in the DNA and we go ahead and convert it into a copy called mRNA, and that this was important because DNA needs to be protected and stay in the nucleus. We can't have it go outside where it can be damaged in the cytoplasm of the cell. So to keep it safe, like a book locked in the restricted section of a library, We have to go ahead and make a pristine copy of it, and that copy has to be able to leave the nucleus and travel out to where the actual building process happens in order to make the proteins. So you can think of the DNA as being the master blueprint we don't want to lose. We have to make a copy of it. The mRNA is the copy that we can go ahead and destroy if we need to, or it can get, stuff spilled on it or whatever. It doesn't matter. And that copy comes out here, and that is what the construction workers use to actually construct the building, or in this case, the protein. So there are different types of RNA. And last time we focused on messenger RNA. There's also ribosomal RNA and transfer RNA, which we're going to talk about here in more detail in a minute. Messenger RNA is going to be the exact copy of the nonsense, or really what I should say is the antisense strand in the DNA. What we also call the template strand is going to, this is going to be based off that, and it's created by RNA polymerase, which goes down and binds onto the promoter and all that stuff, and creates mRNA. mRNA travels out where it's going to meet ribosomal RNA, which is basically the builders. They're going to be the ones actually doing the construction work. And then the transfer RNAs are going to be the helpers to the builders that bring in the building supplies, in this case the amino acids, to construct the protein. We'll talk about those in a little bit. So we then talked about that a gene is actually made out of several different sequences. We have a promoter region, which is going to include an operator where there's going to be binding sites for repressors and things like that. There's going to be a coding sequence, which is the actual gene. This is the instructions to build the building. And then the terminator sequence, which is going to be, of course, where we know where to stop. So our polymerase is able to release and know that, hey, I'm done. That's all that I need to code in the DNA. So let's go ahead and continue. The antisense strand is transcribed. That's the one that is going to be the mRNA. Based off of the sense strand is not transcribed, so we don't use the sense strand, we use the antisense strand. Again, we said this is confusing, and just remember it's backwards to what it feels like it should be. There is a reason for that, we'll talk about it later. Okay, so the process of transcription, we know that RNA polymerase comes down and binds into the DNA strand. We know that certain conditions have to be met in order for it to be able to create a strand of mRNA. Essentially what needs to happen is that whatever substance needs to be broken down needs to be present in order to get rid of the repressor on that gene. I'll go ahead and see if I can go ahead and show, will it show me that here? Alternative splicing, gene expression. Here we go. So we have here our promoter region right here. We need certain factors that are going to be able to bind to the promoter. So repressors are going to be things that are going to stop the RNA polymerase from going ahead and creating the mRNA copy of that gene. So the repressor needs to be removed first. Activators can be added in order to enhance the speed of transcription, or basal factors are just going to bind to the promoter and keep production at normal. So basically these are the on and off switches that are going to make it so the gene is either made or it's not made. And remember, it's important to keep genes off until we need them to conserve energy, just like you keep lights off to conserve energy when you're not in the room. Once we go ahead and create the mRNA strand and the RNA polymerase detaches from the DNA, we then go ahead and splice out all the introns that are within that segment. So these are basically non-coding regions, and we splice them out in such a way and splice together certain exons so that we get an mRNA, a completed mRNA. Think of it this way. We made a copy of the blueprints, and then we go ahead and edit them down to just the bare essentials of what we need to know, and then we send them out. So we basically make those little modifications. It's called post-transcriptional modification. It happens after transcription, and it modifies the mRNA so that we just have a set of instructions that are completely edited and ready to go. Okay, when that happens, we can have alternative splicing. So certain exons can be stitched together in different combinations, and that can actually change the structure of the protein that we're going to make. You can think of this as last-minute edits by the architect to say, hey, in fact, we don't have the building supplies for this, or they don't have the money for that, or they wanted to change this one feature, and so they go ahead and make that slight change. In your body, that context is going to be that maybe there's a certain scenario that's arising and you need a certain shape of protein. That's what your body's going to do. And you're going to go ahead and express that gene. So basically the mRNA is going to go out into the cytoplasm. It's going to start the process of translation, and you're going to make that protein based off of that RNA. Okay, we talked about acetylation and methylation. This is that epigenetics and stuff the environment can affect your gene expression. Cut genes on and off. So now let's go ahead and go to the new content. So translation. So we've come all the way from the nucleus where the DNA has been replicated. It's been transcribed. We have a copy of mRNA. Now what do we do? Okay, so we have our transcription. It's all done. Now translation. So we're going to start with the ribosomes. Ribosomes can either be floating free in the cytoplasm, or they can go ahead and be already attached to the endoplasmic reticulum, or the ER, the rough ER specifically. Basically, ribosomes are little factories, and they're the sites where proteins are actually made. They are made partially of protein and RNA, so they're a combo. They have two subunits, one large subunit and one small subunit, and the two fit together into this nice little two-part piece. The large subunit contains three binding sites called A, P, and E. So APE, yay! In ribosomes, again, exists freely in the cytosol, which is the cytoplasm, or are bound to rough ER. So let's go ahead and continue. Transfer RNA, which is the other type of RNA we haven't really talked about very much, is going to carry amino acids to the ribosome. So if we thought about this in terms of an analogy, I like to think of the mRNA, which is this right here, based off of the DNA. I like to think of the mRNA as being a copy of the recipe you want to make. I like to think of the ribosome as being the cook, and I like to think of the tRNA as being the helper in the kitchen that brings the cooking supplies to the cook so the cook can put it together to a meal. So if we're thinking about this in terms of your kitchen, maybe this is the copy of a recipe that your mom or dad or guardian brought in. You go ahead, this is them making the meal, so your mom, dad, or guardian is making the meal, and you're in the kitchen helping by bringing in kitchen supplies to them, and maybe they say, hey, bring me the flour from the cabinet, and you go get the flour from the cabinet and you bring it over. So transfer RNA have 4 key regions. We have the acceptor stem, which is going to carry that amino acid, the anticodon, which is going to latch onto the mRNA to make sure that it's adding the right ingredient at the right time. Basically, it's like asking your mom or your guardian or your dad or whatever to just be like, hey, is this the right thing? A T-arm, which is going to associate with the ribosome, so it's going to kind of be you coming up and putting your arm on the cook and being like, hey, you okay? And a D-arm associated with the tRNA activating enzyme. So basically this is going to, I don't know if there's a good analogy for this. It's basically going to activate the tRNA. It's called tRNA obviously because it's a T shape. So tRNA activation, not really, don't really need to go into this too much because this is more of an IB style thing. Basically, the amino acid has an ATP added to it, and that phosphorylates it. So we end up having amino acid connected to what we call AMP, adenosine monophosphate, which basically means we got rid of 2 phosphate groups. So when that attaches, it goes ahead and attaches onto the tRNA, and it's ready to go. It's ready to be carried over to the building site. But again, it's a lot. Okay, so translation. Translation is the process wherein we take that mRNA that we got from transcription, and now we're going to go ahead and take that copy of the blueprints or recipe or whatever you want to call it, and now we're going to go ahead and make the meal or make the building. So we're going to use that to build something. Okay, so we take that mRNA and we're going to go ahead and start with a process called initiation. Okay, so basically the ribosome is going to clamp down on the mRNA and it's going to hold onto it and be like, okay, so we're ready to read. Elongation is going to be where the ribosome reads down the mRNA and it's going to start producing that protein. It's going to start kind of pooping it out, one amino acid at a time, and kind of put it together. So it's going to be like a long growing strand coming off the RNA. Translocation is when the ribosome is kind of shifting down the mRNA. So I like to, it's like an old-fashioned typewriter where the the ribosome is on the mRNA and it's kind of sliding down. And as it's sliding down, it's reading and kind of typing out this long polypeptide, this long string of amino acids that will eventually turn into a protein. Then we have termination, which is going to be where the ribosome is going to detach from the mRNA because it's done with its job, and then it's going to go and attach to a different mRNA later on. Elongation and translocation are repeated over and over as the ribosome moves along the mRNA. Okay, so let's take a look at initiation. So once your mRNA leaves the nucleus, it's going to go out to the cytoplasm, and the first thing that's going to happen is the small subunit of the ribosome is going to be attracted to the mRNA. Specifically, it's going to be attracted to this little, well, here it says AUG is here. It's interesting. I would have put it over here, but whatever. AUG is here. It's a start codon. We're going to talk about that in a second. So it's going to go ahead and bind onto that here at this little, what we call the start codon, and the first tRNA is going to come down and bind onto that as well. So basically we have those little instructions come out, and we're going to have someone come up and grab the instructions and be like, aha, okay, here's the fresh instructions from the nucleus, let's begin. And that person's going to call over the first little helper to bring the first step of the building supplies. When that happens, the large subunit clamps down and the cook or the builder, whatever you want to call the ribosome, is ready to go. And so when it comes down, it's going to make sure that the first little tRNA here is slotted into the P slot. You can see EPA. Just, you know, like the organization EPA. And so the, sorry, what we can see here is that the P is the initial starter site for this. So basically, let's recap. mRNA is the instructions copied from the DNA. It leaves the nucleus, it comes out to the cytoplasm. It is attached to the small subunit of the ribosome and the first tRNA. The large ribosome subunit clamps down onto it, making sure that the tRNA is in the P position, and we are ready to begin. Now what's going to happen in elongation is the ribosome is going to shift down the mRNA, and as it shifts down the mRNA, it's going to read the next little piece of mRNA, and it's going to ask the next tRNA to come down and bind on. The next tRNA binds on, and the chef, or the ribosome, goes ahead and puts together the two amino acids being held by the first and second tRNAs in a peptide bond. So these two guys are going to become attached. Okay, and this is what we call elongation. This continues and continues. So basically the tRNA shift over, the one that was in the P slot goes out into the E and is ejected, and another one comes down into the A, and we end up getting a long chain of amino acids formed. So you can imagine this occurring over and over and over again, right? We add another one, we go ahead and get rid of that one, we add a third one, and this long chain of amino acids keeps growing. That chain of amino acids will eventually turn into our protein later on when it's allowed to fold. Okay, so this is what we call elongation, as we are elongating this long chain of amino acids. When we're done elongating, we get to termination. Termination is basically where a termination factor, which is what we call a stop codon, is detected. And the termination factor or release factor comes down and binds onto the A site. And when that happens, instead of pushing the P over, it goes ahead and releases the finished polypeptide, all these amino acids together. It releases those, it releases the mRNA, and it releases the ribosome and everything else. So basically everything kind of just explodes apart, and we're left with what we wanted, which was this polypeptide sequence. So that's termination, basically just stops the whole process. Okay, so I want you guys to look carefully at what happens here. Wow, that is not what we want. Sorry, that was very loud. So basically what's going to happen is we're going to start with our mRNA. Okay, it's going to go ahead and attach this first little piece of our RNA. It's going to go down to what we call the start codon, and the rRNA is going to lock on. The next tRNA is going to come down, and as the ribosome, or rRNA, reads down the mRNA, it's going to keep recruiting these tRNAs, and the amino acid chain is going to start growing. That's elongation. Eventually, it will reach a stop codon. And when it reaches the stop codon, it's going to go ahead and eject everything, and we're going to be left with just a completed polypeptide, just a bunch of amino acids connected by peptide bonds. Now, it's possible for more than one ribosome to attach onto the same mRNA strand. So we're actually going to see here what we call a polysome, is when a bunch of our rRNAs, or ribosomes, sorry, I keep using that terminology, but ribosomes are also called rRNAs. Ribosomes are going to attach onto this mRNA strand, and a bunch of them are going to be sequencing sequencing proteins from this. Now, it's very important and when your body really needs a protein, like say you drink a lot of milk and your body really needs to produce a lot of lactase to be able to break down that milk, that's where this is going to come in. So basically you're going to have a bunch of ribosomes attaching onto the mRNA and making as much lactase as possible in order to help you to break down the milk. And the lactase is this polypeptide that's coming out, and it'll fold into the lactase shape and be ready as a full-fledged enzyme. Okay, so when we think about this, after proteins are constructed using translation, they're going to go ahead and travel to wherever they need to be. If they are the product of free ribosomes, they can just go ahead and go wherever. But if they're product of bound ribosomes on the rough ER, they're going to be sent through the rough ER to the Golgi body, and they're going to undergo some more physiological changes to their structure. Basically, they're going to be edited a little bit before they're sent out. Think of it like this, like a car assembly line. The factory builds a bunch of cars. When the cars are done here, the bound ribosomes, they're going to be sent through the endoplasmic reticulum where they're painted, they're checked, make sure the brakes are working, make sure all the internal systems are good before they're released into wherever they need to go in the cell. So all of this is going to be a little bit above what we need to talk about, but I'm going to briefly go over it because we have enough time, and I think it's an important thing to realize. So how do the cells actually, how do the ribosomes actually know whether or not they should be, free-floating or attached to the endoplasmic reticulum? So if a particular protein is destined for the rough ER, there's an actually specific amino acid sequence. And so what's going to happen is as they start to begin the process of translation, the first thing they're going to make is that little sequence. And that sequence is going to come down that sequence is going to be detected, excuse me. So basically we have our mRNA, the ribosome reads it, produces the sequence is detected, and then the ribosome is locked into the ER so that when it produces the protein goes immediately into the ER. Okay, once inside the ER, the protein is allowed to, or the polypeptide is allowed to fold into a protein, right? So it goes to the primary, secondary, tertiary, and quaternary sections that we've already talked about before when we talked about proteins. And this fold is going to determine the protein's function, right? Because we know that the shape of the protein determines what it does. And so that primary structure is just going to be, right, those amino acids that are all together. So this is the long chain, this polypeptide is what we call it, produced by the ribosome. Secondary structure is an alpha helix or a beta sheet, just basically ways in which these fold. But these are formed between bonds and associations within the base structure of the amino acid, not its side groups. The tertiary structure is the final fold of a protein that's based off of the interactions and bonds between side groups. And then we can have, of course, quaternary structure, which is more than one protein actually bound together to make a super protein, right? Going back here, we can have globular proteins that are going to be soluble in water. Usually they're going to be sort of three-dimensional and blobbish in shape. And over here, fibrous proteins are going to be things that are going to be important to not be soluble in water. So the cytoskeleton of your cell is made out of fibrous proteins, your muscle fibers, all that kind of thing. Not good if they could be dissolved by water. So that's basically it in a nutshell, okay? So again, just going back really quick just to make sure we understand what we talked about. Okay, and I know the slide that I'm looking for is right here. So we started with DNA in the nucleus. We replicated it. We had a good copy of it. It was good to go. We have that DNA. That DNA contains segments of DNA called genes, and those genes are going to be instructions for making particular proteins that our body needs to use. Okay, so when that needs to happen, the DNA is going to be uncoiled and unwound by all the proteins that you guys have already learned about, and it's going to be copied using RNA polymerase and transcription into an mRNA strand. And this is going to be a copy of the instructions in the gene and DNA that allow us to be able to create a protein. That mRNA strand is then going to associate with ribosomes, which are going to go ahead and lock onto the mRNA strand, and they're going to start sequencing long strings of amino acids called polypeptides. And when it reaches a stop signal, it's going to go ahead and detach from the mRNA, and those long strings it made are going to go ahead and fold into proteins and produce the final product. The proteins will then be able to make whatever structure they need to in your body, or they're going to be able to react to whatever is going on within your system, right? So if you've eaten something recently that requires an enzyme, they're going to go ahead and help to chew up that food. All right, guys. So that's it for translation. We have now gone through replication, transcription, and translation. Next class, we're going to go ahead and talk about the control of this stuff, cell-to-cell communication, and how signals can be sent between your cells to initiate this process. So essentially how one cell can look at another cell and say, hey, this is going to happen, you need to produce a protein right now. And they're going to send the signal to that cell, and it's going to kickstart the process of transcription and translation on a particular gene to produce a protein that that cell needs. It's the equivalent of you calling up your neighbor when there's a storm coming and saying, hey, now is a great time to go to the store, or hey, I have supplies for you just to warn you this is going to hit soon and I want you to be prepared. All right, guys, I hope you have an absolutely wonderful day. Thank you so much for sitting through this. I know it's a lot. Just one step at a time. We're going to nail this exam. Keep practicing. Keep going over Khan Academy, keep making sure that you're attending the FRQ practices and watching the videos if you can't attend. It's going to be great. You guys are going to do awesome. Okay, I hope you all are doing well and staying safe and healthy. I really miss you guys, and I can't wait to see you again, hopefully very soon. All right, take care.